Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied about plasma membrane and membrane proteins. In today's session, we will study the transport mechanisms. The permeability across substances across the cell membrane is dependent on their solubility in lipids and not on their molecular size. Water soluble compounds are generally impermeable and require carrier mediated transport. In this diagram, we can see that non lipid soluble molecules require carrier protein, whereas lipid soluble molecules are transferred directly across lipid membrane. An important function of the membrane is to withhold unwanted molecules while permitting entry of molecules necessary for cellular metabolism. In this diagram, we can see that water-soluble molecules are held outside cell membranes because water is lipid insoluble. Transport mechanisms are classified into first, passive transport, within which there are simple diffusion, facilitated diffusion, and ion channels that are specially specialized carrier systems. They allow passage of molecules in accordance with their concentration gradient. Then the second one is active transport, and third one is pumps that can drive molecules against the gradient using energy. Solutes and gases enter into the cells passively. They are driven by the concentration gradient. The rate of entry is proportional to the solubility of that solute in the hydrophobic core of the membrane. In this diagram, we can see that lipid soluble molecules are directly transferred across cell membrane. Simple diffusion occurs from higher to lower concentration. This does not require any energy. However, it is a very slow process. Coming to the facilitated diffusion, this is a carrier mediated process. Important features of facilitated diffusion are the carrier mechanism could be saturated, which is similar to the Vmax of the enzyme. Then structurally similar solutes can competitively inhibit the entry of the solutes. In this diagram, we can see that water molecules are carried through the water channel protein. Then the third one is facilitated diffusion can operate bidirectionally. Then this mechanism does not require energy, but the rate of process transport is more rapid than simple diffusion process. The carrier molecules exist in two conformations. These are ping and pong states. In the pong state, the active sites are exposed to the exterior when the solute binds to the specific sites. Then there is conformational change. In the pink state, the active sites are facing the interior of the cell, where the concentration of solute is minimal. This will cause the release of solute molecules, and the pink and the protein molecule reverts to the pong state. By this mechanism, the inward flow is facilitated, but the outward flow is inhibited. In this diagram, we can see that carrier protein showing ping and pong state. Hormones regulate the number of carrier molecules. For example, glucose transport across membrane is facilitated diffusion involving a family of glucose transporters. Aquaporins. These are water channels. They are a family of membrane channel proteins that serve as selective pores through which water crosses the plasma membrane of cells. They form tetramers in the cell membrane and facilitated the transport of water. They control the water content of the cell. In this diagram, we can see that water molecules pass through the aquaporin channel across the cell membrane. Diseases such as nephrogenic diabetes insipidus is due to the impaired function of the channel. Channelopathies are a group of disorders that result from abnormalities in the protein forming the iron pores or channels. A few examples are cystic fibrosis, which involves chloride channel, then Liddle syndrome, which involves sodium channel, and periodic paralysis, which involve potassium channel. Before moving further, I want to tell you that please like, share, and subscribe the channel. Your one click on the subscribe button shall motivate me to prepare further videos. Any suggestions can be given in the commentary box. Coming to the next, ion channels. Membranes have special devices called ion channels. Ion channels are transmembrane proteins that allow the selective entry of various ions. The sodium potassium pump membrane proteins that allow the selective entry of various ions. These channels are for quick transport of electrolytes such as calcium, potassium, sodium, and chloride. These are selective ion conductive pores. Ion channels are specialized proteins molecules that span the membrane. The channels generally remain closed, but in response to stimulus, they open, allowing rapid influx of ion across down the gradient. This may be compared to the opening of gate of a cinema house when people rush to enter in. Hence, this regulation is named as gated. In this diagram, we can see that ion channel, it is opened when messenger is attached to the ion channel. Such ion channels are important for non-impulse propagation, synaptic transmission, and secretion of biologically active substances from the cells. Ion channels are different forms of ion transport pumps described below. Coming to the definition, ion channels is an integral protein that transports ions across this concentration gradient across the membrane passively, whereas ion pump, it is a transmembrane protein that transports ions against the concentration gradient across the membrane actively. Coming to the ion movement, ion channel has passive movement, whereas ion pump has active movement. Coming to energy consumption, ion channel does not require energy, whereas ion pump requires energy. Coming to need for gates, ion channels needs a single gate, whereas ion pump needs at least two gates. Concentration gradient. Down the concentration or electrochemical gradient is seen in ion channel, whereas ion pumps drive the concentration against the concentration or electrochemical gradient. Coming to last types, ion channels have ligand gated channels and voltage gated channel, whereas ion pumps require has primary and secondary active transports. Coming to the ligand gated channels, ligand gated channels are opened by binding of effectors. The binding of a ligand to a receptor site on the channel results in opening or closing of the channel. The ligand may be an extracellular signaling molecule or an intracellular messenger. In this diagram, we can see that the ligand gated sodium channel is open when a neurotransmitter is attached to the channel, whereas it is closed when a receptor binds to it. Acetylcholine receptor is the best example of ligand gated ion channel. It is present in postsynaptic membrane. It is a complex of 5 subunits consisting of acetylcholine binding site and ion channels. In this diagram, we can see that acetylcholine is attached to acetylcholine binding site and the sodium channel opens. Acetylcholine released from the presynaptic regions with the receptors on the postsynaptic region, which triggers the opening of the channel and the influx of sodium. This generally generates an action potential in the postsynaptic nerve. The channel opens only for a millisecond because the acetylcholine is rapidly degraded by acetylcholine esterase. In this diagram, we can see that a presynaptic neuron and a postsynaptic neuron. On the postsynaptic membrane, there are acetylcholine receptors. Coming to calcium channels. Under appropriate stimulus, calcium channel open calcium channels are opened in the sarcoplasmic reticulum membrane, leading to an elevated calcium level in the cytosol of muscle cell. Calcium channel blockers are therefore widely used in the management of hypertension. In this diagram, we can see that calcium channels are open to presynaptic membrane. It is captured by the muscle and action potential is generated. Amelogenin is a protein present in enamel of teeth that has hydrophobic residues on the outside. A 27 amino acid portion of amelogenin functions as a calcium channel. Phosphorylation of a serine residue of the protein opens up the channel through which calcium ions zoom through and are funneled to a mineralization front. The amelogenin is used for the formation of calcium hydroxyapatite crystals. In this diagram, we can see that amelogenin is the enamel of the teeth. Here ends our session. Kindly like, share and subscribe the channel. Thank you.